Our next speaker is Michael Lisanti from Thomas Jefferson University, and his talk is entitled Parasitic, Metabolic Coupling in Cancer, Understanding Oxidative Stress and Autophagy in the Tumor Microenvironment.
Thank you. It's very exciting to be here. I want to thank Aubrey for inviting me and the organizers. And I'm going to just see if I can figure out how this works. Okay, that's great. So today I'm going to try to oversimplify cancer for everybody. And of course I would like all your feedback. But I'm going to present a sort of a new idea that cancer cells are like parasites. And they essentially use oxidative stress as a weapon to extract nutrients from your body. And this is the weapon they use actually is oxidative stress. So essentially they use the same mechanism as aging to steal nutrients. And we think that this is actually the battery that drives tumor growth. So the idea is simple. We should take out the battery, and then we should be able to starve the tumor cells. And here's a simplified model. So we think that this is driven actually by autophagy, which means self-eating. So the idea is that autophagy in the connective tissue, or tumor stroma, results in fuel production. And then there's a net energy transfer from the connective tissue to the cancer cells, which actually remain autophagy negative or become autophagy resistant because they can kick out genes like Becklin, and then they don't undergo autophagy. So this results in fuel production and fuel consumption. So it's as if the connective tissue cells take themselves and they break them down to component parts, like a house, into bricks and mortar and wood, and then give those chemical building blocks to the cancer cells to make more cancer cells. And this is not a new idea, actually. This was first proposed 5,000 years ago by the Chinese, the idea that all disease, and especially cancer, is essentially an energy imbalance. And it's important to note that as we age, our risk of cancer increases every year. And so aging is actually the #1 risk factor for cancer. Just to make this a little bit more complicated, I've added some more details here. And this is actually all based in experiments that I'm going to show you. But I just wanted to give you an overview first. So cancer cells induce oxidative stress in the normal neighboring fibroblasts. And the way they do this now, we figured out, it was just published in August, is they actually secrete hydrogen peroxide. So the cancer cells are directly inducing oxidative stress in the normal fibroblasts, and this leads to autophagy, which provides three things. It provides recycled nutrients to the cancer cells. Also, the ROS that's produced feeds back onto the cancer cells in a kind of bystander effect, and then the ROS actually mutagenizes the cancer cells and allows them to evolve. And so then the whole idea about mutations in cancer, we think, is that those mutations are to give the cancer cells the ability to make more hydrogen peroxide to get more food from their surroundings. And the third thing, which has important implications for drug resistance, is that the free food that they get from the microenvironment keeps the mitochondria happy in the cancer cells, and so they're protected against death. And so when you give a chemotherapeutic agent, if the mitochondria are happy, then the cancer cells don't die. And we also published that in August, basically showing that fibroblasts confer drug resistance upon cancer cells, and we can mimic that with mitochondrial-rich fuels, such as lactate and ketones. So here, we also think this probably has something to do with cachexia. So essentially, we're thinking that catabolism in the body essentially provides nutrients for anabolic tumor growth. And we think that cachexia, which is systemic wasting, actually starts locally as autophagy in the stroma and eventually leads to negative energy balance so that you have systemic wasting. Again, the same idea. There's a net energy flow from your body to the cancer. And this is just one of the experiments I'm going to show you today. This is a co-culture. Here we've mixed cancer cells with fibroblasts together in the co-culture, and you can see we've stained for ROS production. And the cancer cells by themselves don't make much ROS. But after 5 days of co-culture, we see that they've transferred the ability to make hydrogen peroxide actually to the fibroblasts. So initially the fire starts in the cancer cells, and then it spreads to the connective tissue. And we can actually show that. So at day 2, most of the ROS is actually in the cancer cells, but at day 5, which is as shown here, most of it's in the connective tissue. And we can block all of that with a cheap drug you can buy in the supermarket, at least in Italy, called N-acetylcysteine, which is an antioxidant. So N-acetylcysteine actually has an interesting history. It was first shown to be an anti-tumor agent in 1995, but it's not used at all for cancer chemotherapy. We just tested it on my wife's grandmother. In Italy, she was considered inoperable. She had a tumor the size of a grapefruit because she didn't tell anybody about her breast cancer for 5 years. And so at diagnosis, she was considered inoperable. They didn't want to treat her. We gave her this for 6 weeks. It shrank the tumor to the size of a walnut. And she had the tumor excised, and she's doing fine now. So just to ask, what is the status of mitochondria? And this synergizes quite well with Dr. Berridge's talk. There's something called the Warburg Effect. We think it's essentially wrong. We think that Warburg was half right, that tumor cells actually do use mitochondria, and it's the fibroblasts that suffer. So this is cancer cells by themselves, and we've stained here for mitochondria, and they don't really have much. So that confirms the Warburg effect. But we think that's an artifact of culturing cancer cells outside their normal environment, because cancer cells don't exist by themselves in the body. They exist surrounded in nests by fibroblasts. And fibroblasts by themselves have a ton of mitochondria. But if you mix them together in a co-culture and wait 5 days, now we see, actually, that the fibroblasts have eaten their mitochondria by mitophagy, and now the cancer cells have all the mitochondria. Now, this could be mitochondrial transfer, and we do favor that as an idea, but we can also show, essentially, that we can mimic this or phenocopy it with metabolites. So the idea is that cancer cells by themselves are glycolytic, fibroblasts are oxidative, you mix them together, and they completely flip their metabolism such that the fibroblasts are now glycolytic, and the cancer cells are oxidative. And we can phenocopy that simply by giving cancer cells a mitochondrial fuel, which is lactate. So we think that lactate and ketones are the favorite foods of cancer cells. And I'm going to follow up on that. But this is DNA damage also to talk about another part of this hypothesis. And you can see here fibroblasts and cancer cells by themselves don't show many double-strand breaks. But if we mix them together, there's actually a ton of DNA damage. And it's mostly in the connective tissue, which supports the work of Karas-Eng, who showed that most of the mutations actually take place in the stroma. And we can block all of that DNA damage with N-acetylcysteine, which is an antioxidant, or we can block it with alanine, which is an inhibitor of NO production. So we think that basically antioxidants are important for treating cancer. And the cancer cells think so too, because when they create this havoc of oxidative stress, they upregulate their own antioxidants. So the cancer cells are taking their own antioxidants. They upregulate peroxyredoxin to protect themselves. Essentially, they can buffer exactly how much oxidative stress they receive so they don't die, and so they can mutagenize themselves and evolve. And here is our favorite protein, which is caveolin, that we worked on for 15 years. And this is how we started doing the co-culture, because we found it's a diagnostic, and I'm going to show you the data in a minute, but the most important thing is that if you put the fibroblasts together with the cancer cells, the caveolin disappears. You can see these are the negative cells here, and the cancer cells are green. And so we think that the caveolin is destroyed by autophagy, and it's a trigger for more oxidative stress and mitochondrial dysfunction. And we can block the loss of caveolin with N-acetylcysteine, a cheap drug you can buy in the supermarket, or metformin, which is an anti-cancer drug, which also functions as an antioxidant because it's a complex 1 inhibitor. And we've also done it with chloroquine. The same idea, basically, it inhibits autophagy. So the caveolin is destroyed by autophagy. Here we've knocked it down, and loss of caveolin is actually sufficient to generate more ROS. Again, we can block that with N-acetylcysteine. And this has important clinical implications, because here we've taken breast cancer patients, we've stained them for caveolin in the tumor stroma, and we can score them. And based on this simple, cheap test, we can decide who will have recurrence and metastasis. So this is the total patient cohort. You can see it's 80 versus 20. It doesn't matter if you got tamoxifen or you didn't get tamoxifen. In fact, if you did get tamoxifen, it predicts resistance. Here are ER-positive patients, HER2-positive patients, triple-negative patients. We did also patients with lymph node positivity. It's 80% survival versus 7. So you can pick out all the patients that will have the worst prognosis at diagnosis up to 20 years in advance. Those are the patients that really need the antioxidants. And we've done it with other cohorts, with triple-negative breast cancer, which is the most lethal. You can pick out the patients that will have high survival, greater than 75% at 12 years, versus less than 10% at 5. These are the patients with oxidative stress and autophagy in the connective tissue that is feeding the cancer. This also works for DCIS, ductal carcinoma in situ. It's a precursor lesion. 90% of the patients don't recur. 5% recur to DCIS. The other 5% recur to invasive breast cancer. With this pro-caveolin antibodies, we can actually detect that 5% that recurs to invasive breast cancer. So it's involved both at tumor initiation, tumor progression, and metastasis. Also works for prostate cancer. It picks out the patients with a high Gleason score. And we have unpublished data for other types of tumors, such as melanoma and colon. And so we think this is probably a good diagnostic for most types of cancer, because most cancers have a connective tissue component. So we can also show this is functional. Here we've taken fibroblasts that are actually immortalized with H-TERT, so they're telomerase immortalized fibroblasts. We've knocked down the caveolin using an antisense probe, and we can then make the tumors up to 4 times bigger, both weight and volume, without any change in angiogenesis. So essentially, if the fibroblasts are making food to feed the cancer, you don't need an increase in blood vessels. And we can do that further by overexpressing HIF. So HIF, hypoxia-inducible factor, also is a master regulator of autophagy. If we take activated HIF and put it in the fibroblasts, we can co-inject that with cancer cells and make the tumors 3 times bigger. If we take the HIF and put it in the cancer cells, we actually make the tumors 3 times smaller. So autophagy in the fibroblast feeds the cancer, and autophagy in the cancer cells kills the tumor. So we think this also suggests that oncogenes and tumor suppressors are compartment specific. The idea is that HIF would act as an oncogene in the fibroblast to make food to feed the cancer, whereas HIF in the cancer cells would actually cause autophagy and result in tumor suppression. And further evidence that metabolites are important for tumorigenesis. Here we've done xenografts, and we figured out the 2 most important fuels for tumor growth we think are lactate and ketones. And if we give IP injection of ketones, you can see we make the tumors about 2.5 times bigger without any increase in angiogenesis. Lactate, we didn't see what we're expecting, but then we looked at metastasis, and we see there's a 10-fold increase in the lung metastasis assay. And interestingly, this is what we give our patients in the clinic. The surgeons give IV lactate ringers, also known as Hartmann solution in England, and this is actually causing metastasis. And we can show that. Here's the lungs of the mice. So we should stop using lactate because it's not good for cancer patients. And this is just showing you what it does. As I showed you earlier, you give lactate to cancer cells, and they undergo mitochondrial biogenesis. We can then actually take these different cells, with and without lactate, and do gene signatures. And we think this is one way to do personalized medicine. So basically, this is the strategy here. We take cancer cells, we feed them high-energy metabolites. They then develop mitochondria. We make a gene signature, and we can pick out the patients then who have recurrence, metastasis, and poor survival. And those patients should get antioxidants and drugs like metformin, which are mitochondrial poisons. And one outcome of this study was that we also looked at the gene sets that were increased by gene set enrichment analysis. And interestingly, MCF7 cells on lactate look like neural stem cells with a p-value of 10 to the minus 23. Our statisticians at our IBM that work with us, and they did this for us. And then you can see here, same idea with ketones, hematopoietic stem cells. So basically, we can make cancer cells more stem-like simply by feeding them the right metabolites. And then this is an intersection of the 2 gene sets, and if you look at the intersecting genes, they look like embryonic stem cells. We can take embryonic stem cells, we can give them ketones and lactate, we can make the colonies bigger, we can increase the number of colonies, it's shown here. So stem cells in general like mitochondrial fuels such as ketones and lactate. We can also use the gene signature to look at outcome. Cancer cells using lactate having patients, this is all human patient data. You can see that it's associated with metastasis, recurrence, and poor survival. Same thing true for the ketone signature. So basically, we can now take any cancer cell, feed it the right metabolites, make a gene signature, and predict outcome. And this model that I'm talking about actually is not new. It's been proposed 60 to 100 years ago because it's called the lactate shuttle. And it occurs in muscle. So in muscle, you have fast twitch fibers and slow twitch fibers. The fast twitch fibers are glycolytic. The slow twitch fibers are oxidative. The lactate made in the fast twitch fibers is fed to the slow twitch fibers in this lactate shuttle. Same thing occurs in the brain. The astrocytes are glycolytic. The neurons are oxidative. Again, there's a lactate shuttle. And the way this is accomplished is with specific transporters that shuttle the lactate. The MCT4, or monocarboxylate transporter, spits out the lactate in the glycolytic cells, and the MCT1 sucks it up. And the same thing is true in muscle. There's actually a distribution of MCT4 specifically in the fast twitch fibers and MCT1 only in the slow twitch fibers. In the neurons, MCT1 actually becomes MCT2 because it's a brain-specific isoform, but they're almost identical. So essentially, these transporters should actually be compartmentalized in cancer if there's a lactate shuttle. And so here we've looked at human tumor samples. MCT4 is specifically in the fibroblasts, which are then making lactate and spitting it out and feeding the cancer. You can see the cancer cells are completely negative for this transporter. Here you can see the cancer cell nests. They're all surrounded by fibroblasts that are making lactate, which is being shuttled to them by the MCT4 transporter. And guess what? There's MCT1. It's in the cancer cells. So they're sucking up all the fuel. So again, this provides new targets for therapy. And in fact, several companies have already started to make MCT1 inhibitors, although they're using them for treatment of rheumatoid arthritis, which is a chronic inflammatory disease, just like cancer. And we can also mimic this in the co-culture. So here's the MCT4 in the co-culture. It's not in the cancer cells, not in the fibroblasts. You mix them together, you get oxidative stress. Now that's specifically upregulated in the fibroblasts. The fibroblasts are red because of the MCT4. The cancer cells are green. We can block all of that with N-acetylcysteine, a cheap drug you can buy in the supermarket. Blocks all the upregulation of MCT4. We can cut off the fuel supply. And here are some other related biomarker studies. We can also do this with laser capture. We can actually cut out the stroma and make gene signatures. If we do that, we can completely distinguish. So this is just initially antibody staining. And then we can do gene signatures on the caveolin positive and caveolin negative stroma. And we can make signatures that can completely distinguish between these two different groups of patients. And then we can predict outcome based on the genes that are upregulated in patients that lose caveolin. So we can do something very cheaply with an antibody, or we can do the same thing in a very sophisticated fashion with RNA profiling. And all of this has to do with inflammation, because oxidative stress activates NF-kappa B, which leads to innate immune response. And so we looked at that more specifically. We've shown it with reporters, but here we've done it in the co-culture, measuring the actual cytokines. And you can see here, fibroblasts by themselves, MCF7 by themselves, co-culture. So essentially, mixing fibroblasts and cancer cells leads to a very strong inflammatory immune reaction. This is just a few of the cytokines, but it's essentially a cytokine storm. And so oxidative stress is being induced and inflammation is being induced by the cancer cells. So the cancer cells are acting like macrophages and neutrophils, making hydrogen peroxide and inducing inflammation. And I'd like to thank all the people that did the work, especially my wife, Federica Socha. We worked together. Lots of people in the lab, especially Renee Ballet, Gloria Bonicelli. We have lots of Italians. And all of our collaborators, both here, in the US and also in the UK, especially Agnieszka Winkiewicz, who is our pathologist, Richard Pastel, and Tony Howell in the UK, our statistician, Aris Syringos at IBM. And I think that's it. Okay, thank you.
Any questions?
I'm sorry, I can't see with all the lights.
You mentioned a couple of times the use of metformin to damp down this effect. Metformin is the first-line drug for diabetes, and there must be 1 million people out there taking it. Is there any evidence whether if those people have a lower incidence of cancer?
Yeah, there's about 15 studies that have all shown that patients that took metformin have a 25 to 75% reduction in almost every type of human cancer. And metformin is a mitochondrial poison. And so that proves unequivocally in humans that cancer cells use mitochondria.
Thank you. Can you hear me? Thank you, Michael. That was a really interesting talk. You mentioned that autophagy in fibroblasts would be more of a contributor to the cancer, and that it would actually provide the macronutrients to feed it. But in the cancer cells itself, it would promote their own death. I was just wondering what you thought that would mean for autophagy-inducing drugs and their potential. I'm just aware that there are a few that are now FDA approved, and does it come down to how they're administered? If you were to inject them directly into a tumor, would that increase their viability? Would you say this fact sort of takes them off the table at all as
No, I
useful
think,
drugs?
but it's a really important point. So basically there's something called the Autophagy Paradox, which we've resolved and we've published a simple solution for. The Autophagy Paradox is that if you increase autophagy by giving autophagy-inducing drugs, you kill the cancer in vivo. If you add an autophagy inhibitor, you do the same thing. So the entire autophagy field has been extremely confused by this paradox. But what we think is that it's a simple energy transfer. So if you add an autophagy inhibitor systemically, you'll actually inhibit autophagy in the fibroblasts, and so you'll cut off the fuel supply to the cancer cells. But if you then add an autophagy inducer systemically, you'll induce autophagy in the cancer cells and again starve them to death because they can't use the free food. So we can resolve the autophagy paradox and actually use this now to give drugs systemically to patients to either turn on and off autophagy and confuse the cancer. So I think probably the best strategy, and we've actually written about this, is to give alternating autophagy inducers and autophagy inhibitors, and the tumor will be completely confused and it will just die. And we've actually done some of those studies. They're unpublished. We've used rapamycin and chloroquine as our autophagy inhibitors and inducers, and in animal models we see that's the case. Yes?
What recommended dose do you have on NAC?
I take 500 milligrams every other day right now, and we've also started treating some patients who've come to me via the medical oncologist. One patient was my wife's grandmother. Another one had metastatic cancer to the liver from the breast, and I think they gave her 6 weeks to live, and now it's 6 months and she's doing well. And she's taking, again, through the medical oncologist, she's taking a combination of N-acetylcysteine 1 week, chloroquine 1 week, metformin 1 week, and then just keep alternating. And she had a 70% reduction in tumor mass in the liver in a 6-month period, and her tumor markers went to 0, and she loves to ride horses, and she just won 2 blue ribbons in a recent competition from riding. She's about 55. So basically I think we can treat cancer with cheap drugs that you can buy either in the supermarket or with things you can get with a prescription from a family physician.
With respect, that's a bit anecdotal, is it? I mean...
Oh no, it is anecdotal, but we're going to do clinical trials now. We're going to do clinical trials both in the UK.
But those trials have been done. I mean, 6 years ago, you would have been mainstream. Sorry, I'm over here.
What's that?
6 years ago, you would have been mainstream in, antioxidants are a good anti-cancer therapy. Now you seem to be in a minority, and the field seems to be shifting. You didn't really address that.
Well, I think there are other good studies that have been published recently. One was done by a group in Seattle. They published a paper in Science where they overexpressed catalase in the mitochondria by changing its leader sequence. And they showed that that increases the lifespan of mice by about 25%. And now what they did is they crossed that mouse with the most aggressive mouse model of breast cancer, which is the polyomytal T model. And they converted the tumors from high grade to low grade, and they reduced the metastasis in those mice by 13-fold. And that was just published in BMC Cancer, and we wrote a commentary about that. But there are other things I can't talk about that are coming out in Nature soon. And they also support this idea that cancer cells are parasites. Chloroquine, we think, works just like it does in malaria, because malaria is a parasite. It enters cells, induces oxidative stress, and induces autophagy, and then eats the cells from inside. We think the cancer cells do the same thing from the outside by using hydrogen peroxide. So cancer cells are really extracellular parasites. Dr. Berridge also touched on something called canine transmissible venereal tumor. 2 dogs have sex, and this cancer cell allografts from one dog to the other. Basically, this is a metastatic cancer cell that metastasizes from one dog to the other. People in London have looked at the DNA, the nuclear DNA. They showed that this nuclear DNA suggests that this cell actually was from either a wolf or a coyote from 10,000 years ago. And then they looked at the mitochondrial DNA, and they saw that that came from the host. So essentially, this cancer cell is the longest propagated cancer cell we know of. It's 10,000 years old. And the way it survived over the years is by mitochondrial rejuvenation, by stealing the mitochondrial DNA from the host. So the most metastatic and the oldest cancer cell we know of actually uses mitochondria to survive. And now we see from Dr. Berridge's work that that also applies now to xenografts. So I think we're in a good position to take advantage of this new idea and bring it to the clinic.
Last quick question, please.
Just a quick question about the connection between the megalovirus and cancer and GV.
I'm sorry, I don't know about...
Megalovirus usually is considered upregulate the inflammation. MGV, called MGV, megalovirus.
Yeah, I'm not familiar with it, but we think that, basically I didn't show the other data, but we see that all the inflammatory mediators that are upregulated also individually, when added to fibroblasts, induce autophagy. So we think that inflammation is essentially fueling cancer by inducing autophagy in the connective tissue. So that also resolves the idea. I mean, people know that inflammation is associated with cancer, can drive cancer, but nobody knew the mechanism. We think it's metabolically driven by autophagy in the connective tissue.
Okay, one more question.
Have you experimented with dichloroacetate as a interrupter of these mitochondrial processes?
We have, and we've used a model which is a xenograft where we have mixed together DCA and 2DG, and that helps to get rid of the... So we know that the caveolin deficient fibroblasts make the tumors bigger. We can get rid of that effect by combining that with 2DG.
So 2DG being, sorry?
2DG, 2-deoxyglucose. So we think that, you know, you may have to use combinations, but I think that a lot of the drugs we already have would work against this type of metabolism. We've also shown if we mix in catalase. So for example, if you take cancer cells and fibroblasts and mix together and then put in catalase, you'll kill the cancer cells because they can't make the right amount of hydrogen peroxide to get the free food. And we get 80% apoptosis under those conditions. So catalase is probably the most powerful antioxidant known. It detoxifies 40 million molecules of hydrogen peroxide per second. So if anything, I think we don't have the right antioxidants yet.